Good morning, all. Welcome to our official YouTube channel, SRN BGNR Government Arts and Science College, Khammam. Myself, Dr. B. Satish Kumar, Assistant Professor in Biochemistry. In last three classes, we have been learning about the molecular biology chapters like replication and transcription. In the previous video lesson, we are discussing about the transcription process. In this video lesson, I will cover the post-transcriptional modifications that takes place in eukaryotes only. Okay, so now let us quickly cover some of the summarize. Quickly summarize. I will summarize the previous video lesson. In the previous video lesson, we have seen that central dogma of life. Central dogma of life is how the information flows from nucleotide level, that is from DNA level to protein level. So we have the information in the form of nucleotide sequence in DNA molecules, that is in genes. From the genes, in transcription process, RNA transcript or RNA will be formed. We have three different types of RNA molecules, especially in case of prokaryotes, but in case of eukaryotes, we have more than three different types. In prokaryotes, we have messenger RNA, mRNA, transfer RNA, tRNA, that is tRNA, and ribosomal RNA, that is rRNA. So three different types of RNA molecules are there. So here the messenger RNA is the RNA which involves in the translation process, which has the information or code, the genetic code for the synthesis of amino acids. Okay. So here the information flows from DNA level to RNA level, from RNA level to protein level. So from DNA, if RNA is synthesized, that process is called transcription. Transcription is DNA dependent RNA synthesis. DNA is double stranded, RNA is always single stranded. And from RNA, during the translation process, the polypeptide chain will be, the corresponding polypeptide chain will be synthesized. So in the previous lessons, we have learned about the replication process, which is DNA dependent DNA synthesis. Okay, so far we have covered the transcription and replication also. So during the transcription, which is DNA dependent RNA synthesis, DNA is a double stranded DNA where one of the strand will be utilized as a template strand for the synthesis of RNA molecule, and other strand is called non-template strand or coding strand. So that coding strand will have the sequence similarity with the RNA, except T is present in DNA, and at that corresponding place U will be there in RNA, because RNA never use T, thymine, for as a nucleotide or nitrogenous base. And here from DNA template, one of the template strand, a new RNA transcript or an ARNA will be synthesized. So during the transcription, we have an enzyme called RNA polymerase enzyme. In prokaryotes, RNA polymerase enzyme that synthesize mRNA molecules, or all the RNA molecules, all kind of RNA molecules that are mRNA, tRNA, and rRNA. All three different types of RNA molecules from their corresponding genes will be synthesized by the single RNA polymerase enzyme. Okay, so that RNA polymerase enzyme requires template strand and ribonucleoside triphosphates as substrates so that it synthesizes RNA. Okay, for each nucleotide, for the addition of each nucleotide into RNA molecule requires two ATP molecules because we are using nucleoside triphosphate as a substrate and only monophosphate is being incorporated into the RNA. So that two phosphates, that pyrophosphate will be released, and that releases energy which is essential for the formation of phosphodiester bond in the RNA molecule. So now in prokaryotes we have three different stages in transcription. First one is initiation, second one is elongation, and third one is termination. Okay, so in the initiation, the RNA polymerase binds the promoter site and unwind the double stranded DNA, and it starts the synthesis of RNA on the template strand. So that is, those three things happens during the initiation process. And here we have to discuss about the structure of RNA polymerase. The RNA polymerase is having four different types of subunits. It is a multimeric enzyme. Four different types of subunits, but five subunits are there because it has two alpha subunits. Two alpha subunits essential for chain initiation and interaction with regulatory proteins, and beta subunit, it is for chain elongation. In fact, it binds the substrate, and beta prime, that binds the template strand, and the sigma factor, which recognizes the promoter sequence, the specific sequences in promoter site, promoter region. When we look at the structure of a gene, the gene anyway it should have the nucleotide sequence which corresponds to a specific amino acid sequence in the protein, respective protein. So that nucleotide sequence region is known as RNA coding sequence. Okay? So before that RNA coding sequence, it will have a promoter sequence, which is the site where the RNA polymerase binds. The RNA polymerase binding site is known as promoter site. It binds at the promoter site and unwinds the double-stranded DNA at the beginning of RNA coding sequence. And once the RNA polymerase transcribe the coding sequence, it should be terminated. The transcription process must be terminated once it synthesizes a full length of RNA molecule. So for that, it requires a terminator sequence. So for, at the beginning we have promoter, and at the end we have terminator sequence, and in the middle of these two sequences the original RNA coding sequence is there. This is the structure of gene. So when we look at the promoter sequence is essentially having two important consensus or conserved sequences. First one is at -10 region, it is Tata box, also known as a Tata box, TATAAT sequence. So this is a conserved sequence. Conserved means it is present in all the genes, almost all the genes, and all the genes of different types of organisms, from simple to higher level organisms. So therefore this sequence is conserved throughout the evolutionary process. Almost negligible changes we can see when we analyze different promoters from different organisms or different genes. These sequences are almost similar for every gene in every organism. So that is why it is called conserved or consensus sequence. So another conserved sequence in promoter region is -35 region. At -35 region, that is TTGACA sequence, and these two sequences are the conserved sequences which are recognized by the sigma factor of RNA polymerase. So sigma factor binds promoter site at the place of -10 and -35 region. So if there is no -10 region or -35 region, then sigma factor doesn't bind promoter, and here the RNA polymerase requires sigma factor for proper recognition of promoter sequence. So if the sigma factor is there and it recognizes -10 and -35 region, then only RNA polymerase comes and assembles on the sigma factor. So that is, the sigma factor is the prerequisite, and -10 and -35 regions are very, very important sequences in the promoter region. And the first nucleotide is denoted as +1. The first nucleotide of coding sequence is denoted as +1, and in most of the cases that first nucleotide will be A. And so here this is the RNA polymerase, and -10 -35 region, that sigma factor showed in blue color binds to promoter site, and the remaining subunits comes and assemble on the sigma factor, and finally it forms the holoenzyme. Complete RNA polymerase is called a holoenzyme. So 5 subunits is called holoenzyme, and if we remove the sigma factor from the holoenzyme, it is called apoenzyme. When we look at the promoter site of different genes, we can easily find out that -10 and -35 regions are having their corresponding conserved or consensus sequences. So +1 sequence, +1 nucleotide is also shown here. That is, in most of the cases it is A, not in all cases, but it is A for most of the genes. And during the transcription initiation process, RNA polymerase assembles on the promoter site, and the promoter binding is facilitated by the sigma factor. Okay, so once RNA polymerase binds the promoter region, it unwinds the DNA at coding region and start the synthesis of RNA by utilizing the template strand. Once it starts adding 3 or 4 nucleotides in mRNA, the 3 or 4 nucleotide length mRNA is synthesized, then the sigma factor will be released. Then only the RNA polymerase easily moves or slides through the coding sequence. Okay, so here during the elongation, that RNA polymerase apoenzyme without sigma factor that easily moves along the coding sequence, gradually unwind the DNA in front of it, and synthesize or add nucleotides to the growing nascent mRNA. That nascent mRNA gradually emerges out from the RNA polymerase, and at the back of the RNA polymerase, once again the unwound RNA, DNA again get winded. Okay, so therefore local unwinding and rewinding takes place. And in the termination, last stage is termination. In the termination, we have to know what are the sequences present in the terminator sequence, a terminator region, like that of promoter region. In the terminator region also we have a terminator sequence which is a inverted repeat. In case that means inverted repeat means uh, we have the sequence in the coding strand, the sequence in the upper strand. If you look at that AAA, so that same AAA is present in the opposite strand only, but that is placed a little bit away from this sequence. So the same sequence we will encounter it in the opposite strand. In both the strands we will have the same sequence placed a little bit away. So these are inverted repeats. When these inverted repeats are transcribed, the mRNA region which synthesized from this terminator sequence will have self-complementary sequence. So due to that self-complementary region, they base pair the sequences base pairs with each other and form a stem and loop structure. In the stem we will have high G plus C content, so the stem structure is very strong and it is not easy to dissociate the stem structure. And the stem structure is followed by poly U sequence. So therefore that poly U sequence is bound to template strand with very less strength, binding strength. So these two are the features of terminator sequence. First one is inverted repeat, so that a stem and loop structure will be formed in the mRNA, that is followed by poly U sequence. Okay, so here we have GC rich stem and loop structure and followed by poly U that interacts with poly A in the template strand. So in the termination we have two termination processes. First one is row independent termination and second one is row dependent termination. In the row independent termination, simply the stem and loop structure is formed once the RNA polymerase transcribe the terminator sequence. So in that in at that time the stem and loop structure which is formed in the RNA, that will physically obstruct the movement of RNA polymerase. It obstructs the further movement of RNA polymerase, so that RNA polymerase movement will be stopped and it will try to it will force the RNA molecule and try to move forward, and during that force or struggle, the RNA, the nascent RNA, the newly synthesized RNA is released from the RNA polymerase, because the attachment between the RNA molecule and template strand is very weak due to the presence of AU bonds, poly U and poly A sequences. So these interactions are very weak. During the struggle, that dissociate and comes out easily. So this is known as row independent termination. So we require hairpin like stem and loop structure and followed poly U sequence, and it requires assistance of no proteins like rho factor, and the second termination mode of termination is rho dependent. So rho is it is a protein like helicase which slides over the nascent RNA. It will slide on the RNA molecule by catalyzing, by hydrolyzing ATP and utilize that energy for the sliding purpose. So in this case, RNA polymerase reaches the termination sequence, and once it reaches the termination sequence, it forms stem and loop structure. So that will uh, slow down the movement of RNA polymerase. So by that time, the rho factor will be loaded on the newly synthesized mRNA, which is just emerged from the RNA polymerase. So it quickly moves on the mRNA molecule or nascent RNA molecule by hydrolyzing ATP, and quickly slides and reaches mRNA polymerase. So once it reaches RNA polymerase, it will drag that RNA, pull out the RNA from the RNA polymerase. So here the dissociation of RNA from RNA polymerase requires assistance by rho protein. So that is why it is called as rho dependent termination. So this is the overview of transcription. During the transcription, RNA polymerase binds to the promoter region by the help of sigma factor, and once it binds, it locally unwinds DNA at coding region, and it starts synthesis of nascent RNA molecule starting from place 1 region, and it gradually moves forward, and the RNA will be synthesized based on the template using the substrate molecules. So as it reaches the termination signal, so the RNA synthesis process will be terminated either by rho dependent or rho independent mechanism. So this RNA molecule, which is synthesized by transcription process, is now known as RNA transcript. It may be a mRNA molecule or a tRNA molecule or an rRNA molecule. So far I have explained you about the entire transcription process that takes place in prokaryotes, and now we are going to learn about the post-transcriptional modifications. In fact, these post-transcriptional modifications takes place in eukaryotes only. There is no such kind of post-transcriptional modifications in prokaryotes. So because of that, so that means here, suppose we have a prokaryotic cell, that is a bacteria. In bacteria, there is no nucleus. The DNA molecular entire genome is present in the middle of the bacterial cell. Of course, it is confined to a specific region, but there is no separate envelope or membrane bound structure like nucleus, which we see in eukaryotes. So there is no structure covered around the genome. So this is not there. So that is why it is a prokaryotic cell. So because of this, the DNA is actually exposed to the cytosol. So the translation takes place in the cytosol. Translation takes place here in the cytosol, and here the transcription, replication, everything takes place on the genome. So suppose that mRNA is being synthesized by transcription process in the in this region, that genomic region. Okay. So then what happens, that mRNA will emerge out from the RNA polymerase. So that mRNA will be exposed to the cytosol. So now the translation machinery comes and binds to RNA. So suppose this is the DNA and this is RNA polymerase. So as the RNA polymerase moves forward, the mRNA will come out. So here we have 5 prime and here will be the 3 prime. So as it comes out, the ribosomes binds this RNA and start the translation process. So that is why this transcription and as well as translation, both processes in prokaryotes are coupled. They are coupled. So that means it takes place at the same time. Simultaneous transcription and translation takes place. So before the completion of just during the transcription process, as the mRNA comes out, gradually comes out from the RNA polymerase, then at the same time the protein synthesizing machinery binds to the mRNA and starts synthesizing proteins. So therefore transcription and translation both takes place almost at the same time. So that is why these two processes are coupled with each other in prokaryotes, but this is not the case in case of eukaryotes, because in eukaryotes we have a specific nuclear envelope that surrounds the genomic DNA, complete genome. So the genome is covered under or encapsulated under the nuclear envelope. So that is why inside the nucleus, the true nucleus, inside the nucleus, two processes take place. First one is replication takes place. Second one is transcription takes place. So transcription also takes place in nucleus. So now the translation takes place in cytosol. So therefore, because of the presence of that nuclear envelope, the transcription and translation are not coupled, they are separated. So during the gene expression, the transcription first takes place inside the nucleus, that synthesize a mRNA molecule. That mRNA molecule travels into the cytosol, and where that mRNA undergoes translation process to synthesize its corresponding polypeptide chain. Okay, so in this case the mRNA molecules are really complex. In case of eukaryotes, they require some special modifications after their synthesis. So that is why in eukaryotes the mRNA undergoes some of the modifications inside the nucleus. So that is why another process also takes place in the nucleus, that is post transcriptional modification. So in the nucleus, replication, transcription and post transcriptional modifications takes place. So once the primary transcript, so that is why that RNA is called as primary transcription, it is not the mRNA. So mRNA is that molecule which is formed from the primary transcript after taking post transcriptional modifications. Okay, primary transcript will be synthesized in the transcription process, and that primary transcript undergoes post transcriptional modification, and after that modifications only that primary transcript is called as mRNA. So now the mRNA travels into the cytosol and participate in the translation process for the synthesis of corresponding polypeptide chain. Okay, so therefore whatever we are going to study, the post transcriptional modifications in this video lesson are the eukaryotic modifications. So this is about the eukaryotic post-transcriptional modifications. So we have three different types of modifications. First one is 5' cap, 5' capping. Suppose this is the mRNA or primary transcript. Precisely, it is primary transcript. The primary transcript is having 5' and 3' end, and this first one is 5' capping. So that means a cap will be formed at the 5' region, and poly A tailing, a sequence of poly A, several A's will be formed at the tail region. And splicing means that eukaryotic mRNA contains some coding regions, some non-coding regions, okay, alternatively. So that is why it will have exon, then intron, and exon and intron. So this way it will have different sequences. Exon is the coding sequence which codes the amino acid. Intron which is non-coding sequence, and then followed by another exon and another intron. So these introns must be removed from the primary transcript. Then only the exons will be joined together to form a continuous amino acid sequence in the polypeptide chain. Otherwise, introns will be translated, that results in the formation of some inactive or non-functional polypeptide chain. So introns are non-coding region that never contribute in the amino acid sequence. So that's why these introns must be removed and exons must be joined together. So therefore this removal of introns and joining the exons is known as splicing. So now we will discuss about these three mechanisms of post-transcriptional modification. So first one is 5' capping. So 5' capping means at the 5' end of RNA we will have the 7-methyl GTP cap. Okay, so suppose this is the RNA, this is the primary transcript. In the primary transcript, the first nucleotide will always have triphosphate. The first nucleotide will always have a triphosphate, nucleoside triphosphate. So the first in the first step, during a series of steps, the 5' cap will be formed. So during the first step of this series of reactions is the removal of one phosphate. Okay, one phosphate will be removed. Phosphohydrolase will remove one phosphate, and then from the GTP only GMP comes and binds to the diphosphate, N-diphosphate, nucleoside diphosphate. So we will have guanosine triphosphate here, guanosine triphosphate. So here we can observe that this guanosine monophosphate GMP is interacting with the nucleoside diphosphate. So this interaction is phosphate interaction. In fact, in RNA formation or DNA formation, there always forms a phosphodiester bond between the 3' hydroxy and 5' phosphate group. But here it is not taking between the 3' hydroxy and 5' phosphate. It is the interaction is taking place between 5' phosphate of the guanosine monophosphate and 5' of the nucleoside diphosphate. So this is 5' to 5' phosphodiester bond. So that is why it is just completely closing it is not opening the end and it is closing the head of the primary transcript. Okay, so always the nucleoside, suppose this is one nucleotide, it joins like this, so this will be like this. But at the head side, what happens, it comes in other direction, so in this direction. So this is in this way the GMP is closing the head end. Okay, so uh, that is why it is called a cap is closing the head end. So and then that GMP, so the uh, uh, GP, that is GMP shown in green, is again methylated at specific residues. Okay, so this methylation is called cap 1, cap 2 and cap 3. So that in that way 2 or 3 methylation takes place. The important methylation is at the 7th position of the guanine. That is why it is called 7-methyl guanosine, GTP. Okay, so this is what the capping. So in the capping we will get 7-methyl GTP cap at the beginning of MR, at the 5' end of the primary transcript. So that is capping. And second, here if we look at the structure of cap, we will have this is the mRNA, and in fact these two are the phosphates of this first base, and one phosphate and two phosphate, and this is the GMP. Normally this 3' hydroxy must interact with this one. This 3' hydroxy must interact with this phosphate, but that is not happening here. This 5' phosphate itself interacting with the diphosphate of the first nucleotide, so that is why it is a 5' to 5' triphosphate linkage. So this is the guanosine monophosphate GMP, and this is nucleoside diphosphate. And at the 7th position on the nitrogen we have a methyl group, that is 7-methyl G cap. So this is called the structure of 5' cap, that is 7-methyl GTP. And second post-transcriptional modification is poly-A tailing. So at the end of RNA molecule, at the end of the primary transcript or nascent RNA, newly synthesized RNA or primary transcript, it will have a specific signal, this kind of consensus sequence. This acts as a signal, and so there is an enzyme, polynucleotide phosphorylase, an enzyme. So that enzyme recognizes this specific sequence and add several A's at the 3' end of the nascent mRNA. So that polynucleotide poly-A polymerase, that is the enzyme poly-A polymerase, which recognize this poly-A tail signal and add several A's from the ATPs and synthesize a length of poly-A tail at the end of 3' end of primary transcript or nascent RNA. So this is the poly-A tailing. And so these two when we look at these two post-transcriptional modifications, we will observe that 5' is capped by a 7-methyl GTP cap, and then the coding region we have, and polyadenylation signal sequence, it is followed by poly-A tail. So now the final one is the protein coding sequence. This protein coding sequence is having introns and exons. So that introns and exons are removed by splicing mechanism. So suppose this is the precursor mRNA or primary transcriptor or nascent mRNA. So now what happens here, that this is the exon, red color one, and another exon, that brown color. Okay, so now that each and every intron will have some specific signals, and these signals actually what happens, one of the nucleotide A in the middle of the intron, that nucleophilically attack at one junction, at the 5' junction of exon and intron, and then so therefore that the 5' sequence, that is GU interacts with A. So now in the second stage, the 3' end of the first exon interacts with 5' end of the another exon. So due to that interaction, this end binds to this end, will join with the 5' end of the another exon, and consequently that intron will be released out. Okay, so therefore here first we have intron exon junction 1 and junction 2. So this, first of all, due to this A, the first junction, that A attacks at the first junction, so it will be there occurs a split, a break between exon and intron, and now this exon nucleophilically attack on the second junction upon the exon 2. So now these two are joined and intron will be released. This is called splicing. So splicing, in the splicing, the introns which are non-coding sequences are simply removed from the nascent or pre-mRNA, and the exons are joined together to form a functional mRNA. So now that functional mRNA is ready to undergo translation process. So therefore that formed mature RNA molecule, mRNA molecule, now enter the cytosol where translation takes place. So this is about the transcription and post-transcriptional modifications. In post-transcriptional modification, we have 5' cap, that is 7-methyl GTP cap, and polyadenylation, polyaethyling, polyadenylate phosphorylase, polymerase, which adenylate the 3' end of primary transcript or pre-mRNA. So a polyethyl will be added at the end of the pre-mRNA, and third one is splicing, where introns are removed and exons are joined together to form a functional and mature mRNA molecule. So this is about post-transcriptional modifications, and thank you all.